Well, hello everybody, and welcome to today's webinar, A Practical Guide to Spoilage Investigation and Prevention. So this event, brought to you by Food Safety Magazine, is sponsored by bioMérieux. I'm Adrienne Bloom, Editorial Director of Food Safety Magazine, and I'll be your moderator today, so thanks for joining us. Now, our presenters today are Justin Cabrita, Senior Corporate Account Manager at bioMérieux, and Dr. Martin Wiedmann, Gellert Family Professor in Food Safety at Cornell University. Now, before we get started, please take a moment to scroll down and explore the webinar console. You can download handouts, click on the speaker images to view their bios, and submit your questions or comments anytime using the Q&A box, and we'll address as many of your questions as we can at the end. Today's webinar is also being recorded, and it'll be archived on food-safety.com. And now I'm pleased to turn it over to today's presenters.
All right, thank you all very much for coming today. I appreciate it. It's great to be here. So today we're going to talk about how to stop reacting and move to prevention of microbial spoilage, leveraging our solution called SMARTBIOME. So first, let's jump to a quick poll for the audience here. How do you monitor for spoilers today? If you go ahead and select your answer, traditional microbiology testing, rapid or molecular methods, environmental monitoring and trend analysis, or we don't systematically monitor, mainly just react to specific issues or complaints. Okay. Give it a few more seconds here. All right, let's take a peek at the results. Okay, so for split, it looks like environmental monitoring and trend analysis is the top one. Very interesting. Okay, thanks for participating. Okay, so moving back to SMARTBIOME, how do we get to prevention of microbial spoilage? Well, we need data, right? Data that drives decisions made by quality. And spoilage doesn't just impact quality, right? It impacts production, sanitation, operations. Spoilage can impact an entire facility and sometimes an entire organization. Now, metagenomics can be an inherently complex topic, so we make it as easy as possible. And SMARTBIOME can provide you with data about your facility you've probably never seen before. Most importantly, it's actionable. So what are some of the challenges that we can see related to spoilage? Well, bad smells, off flavors, package bloating, customer complaints of all sorts. But we can go deeper. We can look at shelf life extension, raw material screening, and runtime extension, because spoilage tends to be a common thread that can link all of these things together. And what are some of the consequences of spoilage? Well, if you find a spoiler in your product before you release it, that's great, but that doesn't mean there's no cost associated with it, right? You still have to hold the product, investigate, retest, and so on. And then if a spoiled product does reach your consumer's hands, that's never a good thing. Now you have to worry about your brand image, customer loyalty, and you can incur retailer penalties. And then if spoilage goes undetected for too long, now you're losing usable shelf life, and you can end up paying twice, once to store the product and again to replace it. Now, to add to this challenge, well, we all have some sort of a micro testing program in place, right? But was that micro program custom made for the specific and evolving needs of your facilities? And are you testing using the right tools? We might look at faster or more accurate methods, for example. However, the one thing they don't answer, even though they might meaningfully impact a quality program, is are you testing for the right things to begin with? So just a brief background on metagenomics. So when we compare it to genomics, we're looking at the DNA profile of a specific organism. Okay, so something that's already been isolated one at a time. When we look at metagenomics, now we're looking at all the microorganisms present in a given sample without isolating. So the full picture of the microbial community or the microbiome. And metagenomics is what we focus on for SMARTBIOME projects. Now, even if you have some of this data already, you might, right? You might be sending out and getting some metagenomics data back from a lab. what does that usually look like? A PDF report of some kind. You take a look at it and it tells you maybe a list of microorganisms present and maybe some oddly shaped graphs, and you need to interpret it. So you go start researching separately and look at your list of organisms and try to figure out, okay, what could be going on here? So maybe you research and you say, okay, this one looks like it could be the problem, so I'll take some action based off of this. Okay, that's great. Then you go and you send out some more samples, you get another PDF report and another and another. And ultimately, there's really no way to link all this data together. So you're never quite sure, did you take the right actions and mitigate the issue fully, even if it did happen to disappear. So how does the SMARTBIOME solution work? So of course we start with metagenomics, and we import all this data into the SMARTBIOME software for better visualization and ease of use. And then we add our expertise to add deeper meaning. Now, the people at bioMérieux here that do this are great at it, from the bioinformatics folks to those who can walk around a facility and customize a sampling plan to the specific critical control points and risks that they see. And we take all of this information and we compile it together to generate actionable insights. Because what good is all of this data without clear next steps? So we offer this in 3 different levels of service. And don't get too caught up in the naming of all of them. Really, the main difference is the amount of sampling that we're doing for each one of these. There's some software functions that get unlocked that I'll show you later as well. And also we focus on different goals for each of these projects. So 3 different levels of service to accomplish different tasks. So let's start with the first one that we call SPOT. This is the simplest in theory. Finished product only, usually about 10 to 20 samples total. You send us some spoiled product as well as some unspoiled or clean product, and we take a look at it and we compare using metagenomics on both of them. Okay, and from that we can start to tell you, well, what's there, what could be causing the issue. But we can take this a step further and we can move to what we would call an ACT project. So for an ACT project, we're going beyond just finished product now. We're looking more upstream at swabs, CIP rinse water, raw materials, etc., in addition to the finished product. And we do 2 different waves of samples. So that we do a first one, and then based on what we find, we help you to implement and recommend specific corrective actions to take to help mitigate the issues that you're seeing. And then we do a second sampling wave, usually a bit smaller, to confirm that the corrective actions were actually successful. And then lastly, we have what we call a prevent project. So as you can imagine, even more sampling still. Now we're looking at 2 to 4 different sampling waves in about a year's time. And so here with more data, we're able to generate more insight, and we can start to dashboard this information for you and allow you to take action and prevent future risks, stay in control, and maintain an overall healthy factory. All right, so I'll show you some screenshots now of the software for each one of these levels of service. So we'll start with SPOT. So first thing we can take a look at here is microbiome abundance and diversity in my sample set. So in the top screenshot there, you can see this happens to be a Brazilian cheese customer, which is why it's in Portuguese, but the concept is the same. So we're looking at the total DNA sequences per sample. So here you're looking at the top 5 samples with the most DNA sequences present in my data set. And now on the bottom of that screenshot with the bars, you're looking at diversity. So diversity is the number of distinct microorganisms in each sample. So of those 6 samples, you can see the first one has 7 different microorganisms present, and then 7, and then 6, and so on. We can look at frequency and abundance as well. So at the top in that screenshot, you can see frequency. So frequency is how often these organisms are popping up in my sample set. And then at the bottom, we're looking at abundance. So abundance is how much of each of these organisms is there. So for example, we can say that Streptococcus thermophilus is the most frequent organism at the top, but at the bottom, we can see it's only the 3rd most abundant in my data. And then last, we can look at, perhaps most interestingly, adding some more information and insight from all this data, the cause to effect characteristics of the organisms present. So this part is a lot to unpack, so I'm going to zoom in on the next slide for number 3. So imagine that in my data in the SmartBiome software, I want to look at one organism. So I can click on it. In this case, I'm looking at Licinibacillus sphaericus. And there's no need now to actually go outside of the software. There's a database curated for over 3,000 different microorganisms right there. You don't need to go separately and research. When you click on that organism, you're already presented with a lot of information, as you can see. Is it gram positive, right? Ideal temperature ranges, growth conditions, pH. Is it a spore former? Are there any sanitizers that are resistant to it, or any effective sanitizers, particularly against it? Metabolic information, and in the bottom right, you can look at consequences in the matrix. So this is telling us how that organism specifically impacts my matrix of interest. And in the top left, you can see I chose cheese here, but that is a list arrow. So I can drop that down and look at how it impacts other matrices as well. And I can select different organisms and mix and match if needed. In this case, I'm looking at how the organism impacts cheese. So I take a look and I see, can it cause a color change or slime formation? Yes. pH change? Yes. Flavor, texture, odor changes? Yes, yes, and yes. So most likely not an organism that we want present in our cheese samples. Okay, let's take a look at some screenshots from the ACT version of the software as well. So now I can look at transmission routes, so how these organisms sort of move throughout the facility. So imagine that I had a specific microorganism from the previous slide that seemed like it could be an issue. All the characteristics are maybe matching the challenges that I'm facing. Great, okay. I can click on that organism represented by one of the bubbles here, and it's going to show me all the different samples that are linked to it. So imagine now that organism is found in my finished product, but it's also in my filler and my raw materials and so on. It starts to paint a picture of what could be going on. And now remember I said that there's two different sampling waves that we're doing with ACT projects, before and after corrective actions. So I can overlay this data to show where these organisms are also present in my facility on a map and confirm what that map looks like before and after the corrective actions were taking place. And last, I can implement and track corrective actions through a plan that we co-build with you and can put within the software to follow up on. So I have some examples of actions that we would take, like targeted cleaning for these different projects. However, as you can imagine, it probably varies pretty widely depending on the goals that we're achieving for the project. And last, let's look at a prevent project. So now remember, we have 3 to 4 different waves of samples that we're looking at, including all the organisms present, where they were found in the facility, what zones they were found in, their general characteristics, and specifically how they impact various matrices. So it's a lot of information that we take and we run it all through an algorithm, and we can start to generate these risk scores. So here we can see we're looking at cleaning failure, hygiene conditions, and shelf life. And in this data set, cleaning failure and shelf life have a pretty high risk. So this allows us to take action before these issues occur. And then we can actually look at how these risk categories evolve over time. So once again, I have my hygiene, sanitation, and shelf life, and I'm looking at how they're evolving over the course of 3 different sampling waves represented by the vertical lines that you see in each graph. The horizontal lines represent the zone that each sample was found in. So with this customer, we've actually helped them to significantly reduce their risk over these 3 different sampling waves through the corrective actions that we implemented together. All right, let's take a look at a few slides on a case study, just so you could get an idea of what a real-world scenario could look like using SmartBiome. So let's look at a spoilage investigation for fruit juice. Imagine that you are a fruit juice company and you're facing rising consumer complaints because of bloated fruit juice bottles and all the associated costs, as we discussed earlier with that. What are your goals now? Well, you want to determine what is the root cause of the problem. Implement targeted corrective and preventive measures against it. If it's a microbial problem, identify the microorganisms that are responsible and where they're coming from within my facility. So, as you probably guessed, we're going to follow the three-step approach: spot, act, and prevent. I won't go back and explain them each individually again because we went through it, but I'll take the opportunity to note that we don't have to follow this process 1, 2, 3 either. We can start with a spot project for sure and look at finished product and determine, well, what's there, what could be causing the issue. But we can easily just go right to taking that a step further and look at an entire production line or even an entire facility before and after our recommended corrective actions. Or maybe you don't have any issues right now, or maybe you did in the past and you just want to make sure you don't ever have to deal with that again. We could jump right to a prevent project and start dashboarding a lot of data over time and help you stay in control and avoid that recurrence. So here's the spot portion, or the spoilage part, investigation part of that project. So we ran metagenomic analysis on the finished product, and we found highly fermentative yeast species. There's 3 of them there listed in case you're curious. Now these have robust fermentative capabilities. So this means that they can significantly and quickly influence the biochemical composition of the juice. So that means, for example, the balance of sugars and acids and flavor compounds within the product. And as a result, it impacts the flavor profile, the aroma, and the shelf stability of the product. And of course, that's not a good consumer experience to have. Okay, so then we can take that a step further, right? An ACT project. So looking at more root cause and actions that we can take. So now we looked at more critical control points upstream and downstream of pasteurization. So from raw materials all the way through to the filling machines and the finished product. And we found that the root cause was a hygiene failure in the filling equipment itself. So how did the investigation go? Well, first we found that before the filler there were yeast in the in-process product before pasteurization, but not after. So pasteurization seemed to have been working well there. But then we found yeast in the filling machines as well, even after sanitation. And then after the filler, yeast were found in the finished product. So in addition, we happened to find a bunch of different enterobacteria, lactic acid bacteria, and mold in the raw materials, as well as CIP rinse water in the extractors and tanks within the facility. Now, sometimes when you find the main problem, you uncover other risks along the way that you're, of course, happy to take action on while you can. All right, so take a look at the image that you see on the left there. So this is from the software with some additional language to illustrate what we're looking at for each of those bars. But listed vertically along the bottom, you can see all my different samples in the data set, okay, everything. And so above that, there's a bar, and the bar is going to tell me the microorganisms that are present in relative abundance depending on the size of the bar, and a different color represents different organisms. So first you can see, I'm looking at the product with the deviation, so the one with the swelling in the packaging, as well as juice in process before pasteurization, juice in process after pasteurization, juice in the filling machines, the extractors after sanitation. So really the entire process. It allowed us to have a great view of what's going on. And what do we implement as a result? Corrective actions like revised cleaning protocols, revised sampling plans, and targeted sanitation. And preventive actions like raw material and environmental screening, as well as a routine metagenomic surveillance program. So this customer has continued to send us samples each year for several years, empowering them to stay ahead of potential issues. And last, let's take a look at the facility map. So take a look at that before and after view. So we were clearly able to identify and map out the most critical areas in the facility, take targeted corrective actions that were effective to not only address the immediate problem, but also sustainable resolution through the routine metagenomic surveillance program. So we transitioned the customer from a reactive problem-solving mode to a true preventive strategy. And with continuous monitoring, we could anticipate potential issues and then intervene proactively to help keep their facility map looking like the one that you see there at the bottom. To summarize everything, what did the customer observe as a result of the project? Well, we saw a more than 50% reduction in customer complaints within just 6 months of implementing the solution. improved product stability and significantly reduced production losses, and a decreased investigation time by adopting that preventive, data-driven approach with routine metagenomic surveillance. Because if you're regularly generating and compiling metagenomics data, and then you do happen to have an issue, you already have much more information at your disposal for a quick and efficient investigation. Okay, that rounds out the rest of my slides here. So I'm going to do one more poll before I turn over to Martin. So if you wouldn't mind taking a look there and plugging in your answer. What is your biggest spoilage challenge today? Identifying root causes in complex production environments, detecting spoilage early to act before product release, lack of rapid, reliable detection methods for key spoilage organisms, or managing data and turning results into actionable decisions. Okay, we'll give another 5 to 10 seconds or so. All right, let's take a peek at the results again. So identifying root causes in complex production environments seems to be, by quite a large margin, the top issue. Okay, thank you very much for everyone for participating. I'm going to go ahead now and turn it over to Martin.
Thanks, Justin. And I'm going to add, try to add some color commentary and some color to help you better visualize how you can use some of these metagenomics tools to prevent and detect spoilage issues and other relevant issues in food processing. I'm going to go through big picture, 3 parts. I'm going to talk a little bit in more detail about metagenomics tools, then food spoilage in general, and then go into the meat of it, which is really some case studies. So when we talk about metagenomics today, for those of you who've not necessarily sort of followed the technology, we're really not talking about your grandmother's, amplicon 16S sequencing anymore. We have a variety of different tools within the metagenomics toolbox, or what might be called the metagenomics toolbox, to solve these types of problems. We have shotgun metagenomics, which essentially allows you to sequence all the DNA, all the nucleic acid in a given sample. And that can be very helpful where you want to identify more specifics about the organisms present, such as what enzymatic pathways they might have and how they can contribute to spoilage. We then have more targeted methods within the metagenomics toolbox, which we call amplicon sequencing. The one that's been around for a while is where we amplify a part of the 16S gene that is found in bacteria and then sequence that one. That will give us a comprehensive understanding of the different bacterial taxa, so typically genera, maybe sometimes species, found in a sample. It can be an advantage sometimes if we have a lot of DNA present that is not of interest. If you have a product where there might be a lot of DNA of a plant present, etc., this allows us to really target the bacteria, which can be particularly helpful if bacteria are present at low levels. We can target ITS genes. This is what I'm going to call the equivalent. It's the intergenic transcribed spacer. It's equivalent to the 16S rRNA gene. It's a gene found in essentially all fungi, and the primers are conserved enough to amplify most fungi. We can use that when we want to target fungi. A third and final category that maybe even fewer are aware of is that we now have tools where we target genes that are conserved in a group of organisms, such as genes that are found in all Clostridia, obviously important spoilage organisms, all lactics, all aerobic spore formers, to really be able to target a very specific group of organisms to get better resolution, better assignment to a given species, can better make sure we detect these organisms even if they're found in low levels. Now, these tools are not mutually exclusive. We can use multiple one of them, but they really provide us a broader toolbox than what we used to have in metagenomics 5-8 years ago. So let's talk about food spoilage and some of the challenges. Most of you are familiar with the big picture challenges in that food loss and food waste, and just for education, some people differentiate food loss and food waste, with food loss occurring at the producer, so in the field and the processor, and food waste essentially being food that is thrown away, not consumed at the retail and consumer level. Now, food spoilage, microbial food spoilage, can impact all of these, right? We can detect spoilage early enough that it's really food loss at the level of the processor. Some food loss, some spoilage issues, such as slits in cheese, can lead to greater waste in the production of cheese, as we try to cut cheese into nice little blocks, for example. But then a lot of microbial food spoilage will show up in the retail, food service, or consumer level and lead consumers to throw out product and then ultimately potentially complain to a retailer or a food processor. The scale of all of this is staggering. If we talk about billion pounds of products that are subject to food loss and food waste a year in the US. As this graph that's now from a while ago shows, dairy products, vegetable fruit are big ones, but it really goes across all categories, this challenge of food waste. Now, as we talk about business impact, one thing that we feel is particularly important is when we have food spoilage issues that are what I'm called social media worthy, where you will see, Instagram, whatever it may be, pictures, links to spoilage issues. Taste defects are actually typically not that social media worthy. It's not that easy to have a movie or a picture that shows taste defects, but they are important overall. Color defects are very popular. Funny colored cheeses, funny colored meats. We had what I call the summer of no string cheese a while ago, where we had red colored string cheese. All of those are popular and can have significant negative impacts on industry. Rheological defects are a real favorite, and I'm going to show you a movie of that. It doesn't just happen in milk, but it's common in milk. In milk, we have ropey and slimy milk, which essentially means you can do a nice movie like the one I'm going to show you here, where we have the movie that we created in the lab without any brand. But just imagine someone posting a very similar movie of a container of milk that has your brand on it or your product on it, and showing how it flops into another container or in your school lunch like this. And the reality is there are many of these movies around that had significant negative impacts. I'm aware of one of them that goes just like this movie, but has a brand name on it, that literally garnered 4 million viewers on YouTube. And it's a bunch of school kids, a bunch of school teenagers taking a picture with color commentary and nice little noises as they pour this milk out in what's clearly seen as a school lunch environment. So you can, very clearly visualize the potential negative impact on business and reputation that Justin already mentioned with food spoilage issues such as these. Now, it's not just dairy products, right? We can have similar issues with non-dairy products, but dairy products do provide very compelling movies that are social media worthy. So next I'm going to move into some case studies on food spoilage that sort of illustrate and further illustrates the potential uses of metagenomics and something like SMARTBIOME, and really illustrating also the act, spot, and prevent parts of it. So before we start, and Justin sort of alluded to that a little bit, is that one of the big differences between food spoilage and food safety is that we have a much larger diversity of organisms that can cause food spoilage issues. And pathogens, depending on what product category you work in, you got anywhere from 2 to 3 to maybe 5, 6, 7 target organisms. In microbial spoilage, we're talking about hundreds of different organisms. That's why a database like the one that Justin showed is so important, because it's almost impossible for everyone to stay on top of all the different spoilage organisms, all the different types of defects they may cause, and then be able to use that information to reduce food spoilage and shelf life issues. The other challenge, and there was a little bit, I think, reflected in the answers to the questions, is that many of these spoilage organisms are not easily detectable with standard methods. For us academics and for some testing lab, one of our all-time favorites is to invent new media and new conditions to identify very specific spoilage organisms, like apple juice media, you name it, because some of the organisms require the very specific nutrients found in a given food product to grow. So if you put it on standard plating media, you might not find anything and essentially chase your tail for quite a time before you identify a microbe. Metagenomics tools, SMARTBIOME, cut that time tremendously and allow you to detect these organisms much more quickly. And that's, the spot part that Justin described. So if you talk about metagenomics, what are some of the common applications? Identification of spoilage microorganisms that cause color and sensory defects and their sources. We've seen it in fresh meat. I gave you some examples for dairy products. Gas-producing organisms, a major issue in spoilage, whether it's beverages, some of your sugary beverages, condiments, exploding tomato ketchup and other ones. Anyone in that part of the industry knows what I'm talking about, an eternal challenge. But then there are other, maybe more proactive uses, monitoring of fermentations. And we have different types of fermentative organisms that work together, like in many fermentations. How can we monitor that the relative ratio of them stays the same or stays what it should be during fermentation? Can we predict changes in the efficiency of fermentations? Something that can be very important in large facilities is to make sure fermentations run at the same time and the same time frame every time. I cannot, if I'm on a large facility, have my yogurt sometimes take 6 hours and sometimes takes 9 hours, because that makes it very challenging for me to stage the whole process and make sure I have filler capacity available, etc. Metagenomics tools can allow me real-time monitoring of fermentations to get early warnings of when my fermentation times might start to drift and cause operational issues. And then we can use those tools for shelf life extension and runtime extension. And I'm going to give some specific example of that later. So here's the classical, what I call the spot example, where we try to detect specific microorganisms. Very few of these types of studies are published, but this one was published. And it essentially identified Lactobacillus graminis as a dominant member in a set of products, of refrigerated fresh pork sausage products that showed spoilage. And ultimately, once that organism was identified, it could be traced back to the spice blend as the source of the organism. Now, this picture was more complicated, as it usually is that there was also a change in formulation, where they started a treatment with a growth inhibitor, lactate diacetate. It generally changed the dynamics of growth. And now the Lactobacillus graminis, which before wasn't a major issue, and therefore the spice blend they brought in wasn't a major issue, now became an issue. But with some of these tools, we can identify the issue and then identify the source and correct this issue to reduce the spoilage issue. Another example, one of my favorites, this is some work we did with our group here at Cornell, is we had multiple people reach out to us because they found a very odd defect in fluid milk. And the consumers complained that the fluid milk turned gray. And if you look at the right side, you can see a full gallon container of milk that is nice and white. On the right side, you see a container that is half full and is gray. Now, some of the complaints with this sometimes had not a uniform gray color like here, but had what almost seemed like streaks of gray color. There were concerns about someone injecting ink. Maybe this was more of a food fraud, manipulation, even food terrorism thing. And people had a real challenge figuring out what was going on and what caused this weird color defect. In this case, we did not use the formal metagenomics tools, but it shows how we can use them to identify something like this much, much quicker than we otherwise could. And the root cause here was the presence of Pseudomonas. And with some of these tools, we identified Pseudomonas, but because we got the whole genome information, like you would get with shotgun genome sequencing, you could actually identify a pathway in these Pseudomonas that leads to a color, and specifically indigo, which we think of more as a blue, but when you put it into milk, it really causes more of a gray color. The real interesting thing that helped us with the root cause analysis was if we look at the pathway, you can see that multiple oxygens are required to take indole, or more specifically, the precursors from the tryptophan biosynthesis to indigo. So this is a pathway that requires oxygen. That solved one of the big mysteries in this case, which was this spoilage issue mainly seemed to show up either on the surface or when containers started to get consumed and were half consumed. Now it makes sense because we can see we have more oxygen in the headspace. That now allowed these Pseudomonas to not just grow, but have this very specific pathway that causes this gray color. So we actually helped a number of people that could never find out what the issue was with this. Sometimes they thought consumers just made up things, etc., to identify. No, it was a spoilage issue. It was an organism that was introduced in the facility, and provide clear mechanistic information why the specific spoilage phenotype was observed, which then allows someone to trace it back to a source. So the power of shotgun metagenomics very clearly illustrated with this example. Another case study, and we have a couple of them on sort of apple juice and related products, and a common spoilage issue. This was an issue that, again, was initially pursued with classical methods, and it illustrates the challenge. There was shelf life issues, pressure accumulation, bloating, cloudy content of the bottles, but any micro count, standard plate count, yeast mold, lactic acid media showed no growth. And this is not unusual. Usually when this happens, you then have to do in-depth, very specialized techniques, in this case, direct evaluation, using a bunch of different media and incubation periods, incubation conditions to find the organism. And in this case, it was identified as an anaerobe and saccharolytic spore former, but it took quite a while to determine that. With the spot capabilities, this is much, much quicker. You have a spoiled, you have an unspoiled sample. It's a very, very fast turnaround to identify the organism, cutting through all these in-depth, specialized micro techniques that we had to use in the past for this. This one was identified as Clostridium pasteurianum. No bar toxin genes. Now we could characterize it further, identify growth conditions, helps us with control. All of that is now available through a database, as Justin illustrated. And we're able to trace it back to the raw material in this case. So again, you don't need to isolate. You have a database. You don't need to do d-values. You don't need to determine growth at different pH conditions. You have that information with these improved tools, speeding up this process from spoilage to detection prevention tremendously. Another one, again, apple juice with visual defects. This identified a potentially novel species, so we're not just limited to known species. And again, once you have the species identified, you can do characterization that allows you to identify minimum inhibitory concentration of different preventions, move you to prevention. So common issue, metagenomics will tremendously speed this up. Now let's move into these tools to maybe be less reactive to spoilage issues, and then take in that spoilage issues to preventing, to using it for, I think, what I'm going to call operational efficiency. Let's talk about the potential of using metagenomics tools to extend runtime. A common issue, and in our experience, when we used to get these questions, we run 16 hours and can pick an arbitrary time, and we want to run longer. What we usually will do as process authorities with people is say, okay, you got to run a time series You're going to take a sample every 2 hours, and we're going to test it for certain organisms and to identify whether you can keep your production line under control for your extended runtime. Usually endless debate ensues in terms of, well, what do we test for? Do we test for coliforms, Enterobacteriaceae, total plate count, something else? And there's no real good determination of that because it depends really on a lot of details. With metagenomics, this discussion doesn't need to happen. We essentially do the same runtime experiments, collect samples every 2 hours, and then do a metagenomic profile. And what you expect to see if a run is under control is you see very similar profiles over time as shown in this image below. And we're not looking at formal statistical analysis yet, but you can see, it looks very similar if you think the first is 2, the next is 4, the next was 6 hours, etc. Now, the alternative to this is that when you do the same thing, you might see a change over runtime, and that's illustrated in the image on the right side. You could see that the microbial composition changes, and this organism that's shown in green here starts to increase over runtime, which suggests that there is probably a niche of that organism that starts to contaminate maybe your equipment, and then that organism grows, becomes more prevalent over time, and will cause an issue. If your profile shows something like on the right side, then clearly you do not have microbial control over your extended runtime. And now you need to move to the next level, which think of it as ACT. You want to now identify the source of this microorganism. Hopefully eliminate the source. May have to identify some redesign of your equipment, some redesign of your process that then would allow you to run an extended runtime under full microbiological control. The other beauty is if you do this, this will allow you to also determine which organisms start to grow and determine whether there's spoilage issues because you have a database for that. The principle of that to extend shelf life, very similar. In this case, you would monitor samples over shelf life, see which organisms increase over shelf life. So take a sample, incubate at a certain temperature, let's say 6 degrees, over X amount of days, 20, 30 days, whatever your shelf life is. Take samples, identify which microbes grow, and then go back, identify the sources, and then do continuous monitoring. That would allow you and help you in increased shelf life. I'm going to finish up with a few quick examples on how we can use those tools more related to food safety, but maybe not the classical food safety. Let's detect a problem and then do a root cause analysis. Opportunities there is pathogen reduction and prevention of pathogen growth, but then otherwise also how can we use these tools to reduce the risk of recontamination by validating and verifying hygienic zoning or specifically cleaning and sanitation. These ideas have been kicked around for a while, but with tools such as SMARTBIOME, they can really become much more accessible. The important thing in all of these is that it's not just about creating the metagenomics data, but also analyzing them with advanced analytics. We have shown that with some of the modern AI technologies, such as machine learning, neural network, etc., we can actually do metagenomic screening of raw materials and identify when a raw material is different. That indicates a potential problem and is an early warning of potentially food fraud, but potentially food fraud that goes along with a spoilage issue or a food safety issue. We can, with these tools, collect samples before and after cleaning, which is shown on the left side on this picture, which I took from this prior paper. Then if we zoom out on the first two, which is floor before and after cleaning, you can see that the yellow organism is present in both at similar proportions. Now, a lot of detail in terms of how you analyze it. Is it similar percent or is it similar levels? But you can see that a specific type of organism survives. You can define what organism that is. You can use a database to potentially find out whether you may have to adjust your cleaning and sanitation procedures, whether it's different chemicals or more disassembly, etc. It's a great way to identify weaknesses in cleaning and sanitation. This is two papers that actually came from the US FDA, where they used metagenomics and what they call quasi-metagenomics to identify organisms, specific penibacillus, that can inhibit and even kill salmonella. And part of that identification was because they were wondering why they were less likely to find salmonella in tomatoes grown in certain parts of the US. So it's almost discovery of organisms that could be used for competitive exclusion. So with that, I hope I've shown you the sort of wide application range of metagenomics tools, from the well-defined, that's been around for a long time, spoilage, root cause, and elimination, to new opportunities for runtime extension and shelf life extension. Application of those tools successfully to this requires more than lab work. It's not just about sequencing the DNA in a sample that you give it. It requires analysis. It requires databases, so that we don't have to go back, isolate the organism, and spend another 2 months characterizing it, but having access to these types of data that Justin described. With that, I'm going to turn it back over to Adrian to walk us through Q&A. Thank you very much.
Okay, great. Thank you so much, Justin and Martin, for those great presentations. And we have received several excellent questions, and you can continue to submit questions if you'd like while we're doing the Q&A here. But let's jump right into some questions. All right, so let's see. I'll throw one out. And Justin, maybe you want to tackle this one. How would you solve the problem of biofilms when sampling?
Sure. So I think, with biofilms, it's a matter of really figuring out what organisms are causing them, right? And so, they're attaching, and we need to figure out which ones are there so that we can target those specific organisms. So you saw within the SMARTBIOME software, right? It's going to tell you if you find an organism, can that cause biofilms? And so that allows us to start narrowing it down and take more targeted actions against it. So I don't know if Martin has any other comments too, but feel free, Martin.
Yeah, I think it's a really good question, but also an important one, because I think there's what I'm going to call an operational definition of biofilm in the food industry, which means the same organism is there over time, right? That doesn't necessarily mean it's in an actual biofilm, which is one of these thick matrix where can't be reached by sanitizer. It might just be there over time because it's in a place where a sanitizer can't reach it, right? And I think the sort of the spot and then the act will really help us. Where is it found? Is it likely to truly be a biofilm? Or is it just a niche that is not easily accessible? Some of the additional information on, are these organisms that can form biofilm? Is it a Pseudomonas? Is it a bacillus that's likely to form biofilm versus one that is not, can help with that. But it's that combination of mapping it and having the phenotypic information that becomes important. And then help us make decision like, is the solution really to get a different sanitizer that now has biofilm disrupting capabilities? Or is the solution more equipment disassembly? The sort of act part will help you identify where it is and determine which the right solution is to address this, potential biofilm, because now you know whether it's truly a biofilm or just a niche where you can't reach the organism.
Okay, great. Thank you. And there's another question. I just want to clarify this because I think you guys answered it. But another attendee on this topic was saying, can you use metagenomics to identify a biofilm composition and then decide which enzymes to use based on that?
Yeah, maybe Martin, I can turn that one to you.
I think if you take the right sample and it's a biofilm, you can, right? I think, it requires you to actually know it's a biofilm, find the biofilm, sample the actual biofilm, know what organism is in there. And then some of the characteristics that the database gets, you might be able to help you with it, with identifying. I'm not sure whether it can tell you which specific enzymes to use. I think it will help you direct you towards, is it a question of using enzymatic cleaners versus a different types of sanitizer? If you have whole genome sequences, you might be able to detect some resistance genes saying, this organism really, iodine isn't really going to cut it with this one. We need to change to something different. Or maybe it is a question of enzymes. I think the true value then is that you can continue on the prevent after you change your sanitizer, make sure it's actually working. And I think that's where we often fall down, right? We change a sanitizer that we like because it sounds good, it's got enzymatic activity, but then how do we verify that it's actually working?
Okay, great, thank you. All right, let's move on to another question here. So how would you handle non-pathogenic bacteria or fungi that have not been sequenced yet, so not present in databases, that would result in unclassified data reads?
So, I mean, I can tell you that when our team is going through the data, right, we have a team of customer success managers and data scientists that are looking through everything, right? So if something is found that's not in the database, or maybe they don't have like something that's immediately clear as to the root cause or something to investigate, they are also manually combing through the literature to check and see what else could be the cause, and then that information subsequently gets added into the database. So there is also a lot of manual, customized technical work that goes into analyzing the data. So you saw like those kind of three bubbles earlier in the presentation where I said metagenomics, right, computer science, and then our portion with manually reviewing everything. That importing of the data into the SmartBiome software, there's a lot of bioinformatics work that actually goes into it. So I go over it pretty quickly when I'm talking about it, but in reality, there's a lot of manual data review that happens there. And then I'll let Martin make a comment as well.
Yeah, and I think that's where the methodology comes into, right? If you're doing 16S, you get unclassified read. I mean, I can't tell you what it is, but I can often tell you what it is in a larger group. Does it fall into the Enterobacteriaceae? What it is most similar to? Then depending on the similarity, what's the closest organism? I might be able to sort of predict what it might do, saying, the 5 organisms most closely to all grow at low temperature, probably can grow at low temperature. Or there might be nothing close to it, in which case I'm like, yeah, this is not giving us enough. We now either go back to isolate, or we go back to shotgun metagenomics, where we can sequence everything and try to get an idea of, based on the genes present, what does this organism do? And I think for listeners to sort of visualize that, let's give the Pseudomonas example. Even if I knew nothing about this organism, but I have spoiled product that shows gray color, spoiled product that does not, okay, or unspoiled product that does not show gray color, and I find genes that are important for certain pathways, it will still give me that information based on the mechanism. So I think it's somewhat easier sometimes to deal with unclassified organisms or reads when we have shotgun metagenomics. So there's some bioinformatics solutions to it, but then there might be some solutions about just going back and modifying the methods you use.
Okay, great. Thanks, Justin and Martin. Okay, another question for you. Is there any way to eliminate DNA from dead cells, which may come from heat-treated raw materials?
So you certainly can have, right, dead cells, right? It depends on the quality of the DNA, right, whether we're going to detect it or not. But at some point, we can draw some inferences. So think about like sampling before and after pasteurization, for example, right? We might look at the quantity and what's there before and after to help infer whether or not it's alive. So there's never 100% guarantee in that situation. But I would say to keep in mind that we're also not looking at just one sample at a time most often. We're looking at a full picture. So sampling at multiple points in the process that allows us to put the full picture together, not based on just one sample. So it's less impactful if we happen to find something that's dead, and we still use it as a quality indicator of sorts.
Yeah, dead versus alive is again a good question and often raised as a concern. I mean, Justin did a great job describing some of the solutions to it. The other thing is, if it really impacts your decision making, you have other options. You could take a sample and do direct metagenomics and then do an enrichment and see what's there after enrichment. If it's dead DNA, it's either going to go down in total levels because it's going to get degraded, but it's not going to increase. So you can use some of these additional tools to help you whether the signal is likely to come from dead cells or not. On the other hand, and that's important, sometimes dead cells can actually provide you with some very valuable information. So take milk, there's a coagulation issue that's actually caused by enzymes that are heat resistant. So the organisms aren't going to be there, but the enzymes the organisms made will survive. Now, if some of the DNA of the organisms is still there and the organisms are dead, I can say, this sample has high levels of, let's say, certain types of Pseudomonas. Doesn't make sense that it will be at as high levels after pasteurization. Plus, through some different methods, it seems like they're dead. However, it can still tell me that my coagulation issue is probably a heat-stable enzyme that survived the heat treatment. So it almost allows me to sort of deduce a little bit what happened with the raw material in some of those cases. So not always a bad thing to have dead cells.
Okay, great, thank you. And then another attendee is wondering, how long does a metagenomic determination take?
So that depends on the project. Usually for the total project, so not just the metagenomics testing itself, for the total project, it's usually in the 5 to 6 week range. Now that's including everything though, because like I mentioned before, that piece where we're importing the data into SmartBiome does take a significant amount of time and manual data manipulation to make it work well, which is good. We want that as part of the process. The metagenomics piece within a few weeks could in theory be done by itself, but in order to actually put everything in the SmartBiome software, generate customized reports with action plans that you should be taking, and then confirm those actions later, it does take a little bit more time for that reason.
Okay, great, thank you. Let's see, another one is, is there any way to estimate actual levels of organisms using these technologies? Changing numbers are also important in understanding behavior over time. Any thoughts?
So from my view, and I'll let Martin comment after too, from my view, we're looking at basically the number of DNA sequences present, right? So we're looking at things like relative abundance of the organisms that are there. And that does definitely give us an indication, right, of what could be going on and how much of each organism is present. And we look at the SmartBiome software and we can see, if you remember, the different bars, right? And it tells us proportionally how much of each organism is there relative to the other one. And so that we certainly do use that information to determine what could be going on in the sample set.
So maybe a little bit more to, I think some people are used to, sometimes these data from these metagenomics analysis are presented in proportions. Let's say, okay, 50% of your sample is organism A, 50% of is organism B. But you have no idea whether that 50% is a total of 100 or a million cells. It just tells you proportion. SmartBiome does allow you to look at the absolute levels, so that the read, what we would call the read counts in this case, which gives you an estimate of total abundance, not just proportions. Okay, can you 100% connect it to what you would get as CFU per mil? No. Now, we have many that's a general issue, right? Even if we do MPN, which is one enumeration method, versus CFU, which is another enumeration method, versus another method, they don't always correlate one-on-one. But I think it can give you an idea of the relative, not just the relative abundance, but also the absolute levels of these organisms and whether the absolute levels of rates increase, right? Is it 100% accurate? No. Is it going to be as accurate as plating on a super selective media? Maybe not, but it's really different data. But it doesn't mean the insights you're getting are any less valuable.
Okay, great. Thanks so much. We've still got lots of great questions rolling in, so I think what we'll do is extend a few minutes and try to get to as many of these as we can. All right, so next question for y'all. So when you're talking about off odors, is there a possibility of pairing metagenomic data with gas chromatography?
So, I mean, I'll chime in first and say yes. I mean, in theory, not within the SmartBiome software, but it doesn't mean that we can't generate some additional data and look at it side by side with you, in addition to what we're generating with metagenomics and SmartBiome. So we can sometimes, I mean, customers have a lot of additional data that's really not necessarily even relevant to metagenomics. Even things I would think like time and temperature data, right? That sort of thing. We can definitely look side by side and try to draw some conclusions in parallel with the data that we're generating in SmartBiome. And then that can generate a more powerful picture.
Yeah, and I think that often a company might select that. They say, hey, we're going to send you these samples for SmartBiome, but we also, if we have these tools available, do it ourselves because we have it in-house, and it might be normal sensory evaluation with a trained panel. It might be GC, it might be whatever else it is, or you send it out to another lab. So I think there's some value, particularly as you're trying to identify specific issues, to sometimes combine those two and maybe verify or validate some of the information you might get from a database.
Okay, great, thanks. All right, another question, and I think this kind of speaks a little bit to one of the visuals you showed, Martin, in your segment of before and after with cleaning. So the attendee is wondering, how likely is it that clean in place water could cause microbial concerns?
Trying to unpack that question. I think the water itself probably doesn't cause the concern, but it could be an indicator of an issue, and it depends on when, where, and how you sample it. I think there's a lot of nuance in terms of when you collect the samples, etc. Now, if you have, residual standing water because your pipes don't slope enough and it stays there overnight, and then you start operation without correctly sanitizing at that stage, then that could be an issue. So it can give you some ideas about some of your operational problems potentially, and some of your design problems in your system.
Yeah, I agree. It's not just necessarily coming from the rinse water, right? I mean, there are maybe some scenarios where I think cycling it can occur, but really we're using it a lot of times as an additional sampling point to check what could be going on, right? A lot of this equipment can't necessarily be easily opened for swabbing different parts of it, right? So we have to run the rinse water through it, and that gives us a way to test what's going on inside.
Okay, great. Thanks both. Take just a couple more questions here. So back to biofilms. Is there a risk of a false negative when swabbing a biofilm? Is there a possibility that the organisms won't be picked up if you apply insufficient pressure with the swabs?
Yeah, I mean, absolutely. It's not easy to sample a biofilm in some cases.
Mm-hmm.
I think it also depends on the locations where it is. And I think back to Justin, that it depends. You really got to do a comprehensive sampling, right? You might not necessarily detect it in your biofilm sample, but you might detect it in downstream where you have sloughing off of the biofilm. But I think if you suspect there's a biofilm, usually you should be able to do the right sampling. But Justin, I don't know what you have to add to that.
Yeah, I mean, I don't have, I guess, direct experience with that. We could certainly discuss that too in more detail offline with some of our technical team if whoever's asking the question would like to do so. But yeah, it's about getting a larger picture, right, in general of what's going on, right? Large sample set across multiple different parts of the facility. Yes, that could include a biofilm itself, but it's also including upstream, downstream of that to see what is the actual impact in the end.
And maybe go back to what I said before. I think often the problem might not be that you're not sampling the actual biofilm, but the organism is in a place that your sponge doesn't get to, and you need to disassemble before you collect the samples. I think this is this balance between people think it's biofilm, so it's an easily accessible spot. I'm just not removing it. The reality is, based on my industry experience, 95% of the time, the organism is in a spot where you can't get to. And we're not taking the time to disassemble the equipment to get the sponge or get the swab and the sample of where we can get the sample. So I think that's very important to keep in mind in these sampling excursions, is gotta do some disassembly.
Great, thank you. And then one last question here, and there's been a couple questions along this topic. So identifying spoilage in products that have bacteria in the finished product. So for example, probiotics, where good bacteria concentrations are usually over log 6 CFU per gram, or in dairy products where the finished product has bacterial cultures.
I mean, I'll start.
Yeah,
go ahead, Martin. Go ahead.
I think the challenge there is if you do 16S or shotgun metagenomics, the vast majority of your reads are probably going to be these fermentation microorganisms. That's when you then go to your more specific tools. Say, all right, we got spoilage here. Based on what it looks like, let's do a Clostridium-specific amplification. Let's do a Bacillus-specific amplification. And so that we avoid getting a lot of read saturation from these fermentation organisms. That's the approach I think most people will take in that case.
Yeah, we can use specific methods, right, for the sequencing piece, to better target those scenarios. So yeah, as Martin mentioned, like we have some sort of proprietary methods around protocols just for things like lactic acid bacteria or Clostridia that allow us to just focus on those.
Great. Well, thanks so much. That is all the time we have for questions today, but please join me in thanking Justin and Martin for their presentations, as well as our sponsor, bioMérieux. Now, if you have any additional questions, you can email webinars@bnpmedia.com. We're always happy to help. And when you exit the webinar, you'll be taken to a brief post-event survey. We appreciate you taking a moment to share your feedback as it helps us continue to improve our programs. And you can visit food-safety.com for the archive of this presentation and see information about our other upcoming events. So thanks again for joining us. We hope you found this webinar to be valuable, and we hope you have a great rest of your day.